Hi, I'm back here with a continuation of the process of going from a gene on the DNA template, from the DNA double helix, to the finished product of being a protein. The proteins are what are actually going to determine the phenotype. And remember that the genes that are found on the chromosomes are specific sequences of DNA. And we want to look at how the cells eventually are going to display the phenotype. So again, I know I'm dug out an old totally legit picture from my, college days, but we go from DNA to the finished phenotypes in the body. So those processes that we've already talked about are including transcription, the process of making RNA from a DNA template. But now we want to discuss how we go from that decoded message into the specific protein. Remember that the building blocks of proteins are specific amino acids. So we're going to look at, or we have already talked about, the transcription process. We now want to move further into the translation process. Translation is going from a nucleic acid language of RNA to an amino acid language. So the question is, how can we code for the amino acids with only 4 different nucleotides? So essentially there's 20 amino acids that the body uses, and just as kind of a footnote, there's recently been the discovery of at least another amino acid and possibly even 22 total, but we're just going to stick with this number. Either way, it doesn't really matter because we've got 4 bases in the DNA molecule, A, T, C, and G. We've got 4 bases in the mRNA or in the RNA sequences, A, U, C, and G, but we're talking about 20 different amino acids, and you can put those together in almost an infinite number of ways to get a finished product called a protein. So the mRNA codes for the proteins in triplets. So cutting to the chase, there are 3 bases on the mRNA. Those are called a codon, every 3 bases. As you can look, when we talked about transcription, there's a complementary base pairing on how we get a specific sequence of RNA from a specific sequence of DNA. We don't all of a sudden shift to a kind of a random process here. So each one of these codons is going to code for a specific amino acid. Really, the process that you as a student need to be able to complete is to take the mRNA codons and be able to tell which amino acid sequences you are going to put together to make a specific protein. So you would never have to memorize any of this. You would never have to memorize this genetic code, but you have to be able to use this chart. Looking back, I think that since the remake of the AP biology exam, this amino acid chart has been on, I think, every test except one, and I may be mistaken exactly there, but let me tell you, it's a good thing to know. So this code, one of the coolest things is this code is for all living things. In other words, all life, all cells read the same code. That's why it's called the central dogma of life, right? Where we go from DNA to RNA, and then the RNA code is read into a specific order of amino acids, thus giving us the phenotype through protein structures. But we've got a codon. Well, first, to use this chart, we're going to take every 3 bases, remember the codons, and we're going to find the first base here. It's going to be one of these 4. If, let's say that it is U, well, then we know that it's going to be completely in this portion of the chart. The second thing we're going to do is we're going to go up to the second base. And if it's U again, we know that it's going to be in this column. So now we've narrowed it down to a specific row and a column. And thirdly, we need to find the third base And the third base, let's say that it's the letter A, or adenine, so now we know that it's going to be this base. So from that order, first base being uracil, and adenine, we have the specific amino acid that we're looking for. In this case, it happens to be leucine. As you might notice here, this third base wobble really refers to the fact that the code is redundant. You see, and really what the third base wobble means is that really the third base is kind of less important than the first two bases. The first two bases, you notice, really kind of determine whether or not it's going to be a different amino acid in the chart. For instance, here with this base, you can see that there are a number of ways, C being the first base, U being the second base, but then each of the third base, you still get the same amino acid. So that's kind of a little bit beyond our scope, but just so that you know, again, the code is redundant, meaning that there's several codons or triplet bases from mRNA that can lead to a particular or code for a particular amino acid. There's also some special ones. You'll get used to seeing this. AUG, this codon is almost always the start codon. It is the start codon for the process, but it codes for the base or the amino acid methionine. And then there's three different stop codons, and these are the three codons, or excuse me, the three-letter triplet in mRNA that signal the stopping of the process. So the actual reading of the amino acid chart, again, you would never have to memorize any of that thing. You simply need to know how to use the chart. And it always uses the mRNA strand. But conceptually and realistically, what is actually going on in the cytoplasm at the ribosome? And what happens is that the amino acids that we've talked about that are linked together are actually carried on a third type of RNA called tRNA, or transfer RNA. That's because its function is to transfer the amino acid to the growing polypeptide chain. Polypeptide is really just kind of a short building segment of what eventually is going to become a protein. But here are your codons, every three bases, codon, codon, codon. Remember that these are specific from the transcription of a DNA sequence, but on one end of the tRNA molecule is a complementary anticodon. The anticodon is complementary to the codon. Well, the other unique part of the tRNA molecule is that it also carries the specific amino acid that we were talking about. So physically and literally, the tRNA molecule is going to carry the correct amino acid, and when I say correct amino acid, it's because the anticodon is complementary to the specific order of bases on the mRNA strand. Take a second, pause the video, and take a look at the specificity between the codon sequences and the anticodon sequences. This transfer RNA, or tRNA structure, it's referred to often as the clover leaf structure because it sort of looks like this. Of course, it's a three-dimensional structure, but this is a nucleic acid. This order of bases would be transcribed from a DNA molecule as well, but there are two main parts that we need to know about, and that would be the anticodon end, the three bases, and the amino acid that would be bonded to the 3' end of the tRNA. So again, here we are actually at the ribosome, which we haven't really mentioned yet, but we are seeing how we get from the nucleotide sequence on mRNA to the anticodon, which is going to be found on the tRNA, as well as having the correct amino acid with it. Really, any complexity of the RNA in the ribosome, or the specific rRNA in the ribosome, is a little bit beyond this, the scope of our class, but the ribosome has two units. They're called, well, two subunits, a large and a small. And really what that does is it sets up a little, a bonding area to be the location at which the mRNA sequence of codons can then be actually bonded and processed with the incoming tRNA molecules in order to build a protein. In this course, we're not going to need to differentiate between these three sites in the ribosome, the A, P, and E site, but really the mRNA sequence is going to move through those sites kind of as a tick, tick, tick stop. I mean, it's going to move in, it's going to shift over a spot, it's going to shift over a spot, and it's going to shift out. But there's a first, second, and third order for those spots. And again, we don't need to keep those straight, but just so that you know that that's what you're going to be looking at as we dive into the process. So as we've already talked about, we have three specific segments of translation, of building a polypeptide. Remember, the finished product of a polypeptide is going to be a protein, but this polypeptide chain is where we link together the individual amino acids. So the tRNA is going to bind in a specific spot. Of course, we know which one it's going to be. Take a look at it. You've got a complementary anticodon on this molecule that binds with a proper codon here. The initiation is going to be the start of the process. Elongation, much like transcription, is going to be adding amino acids in the specific sequence, depending on the codons. And then the termination, when this whole process is kind of ordered or signaled to stop. But as we bring a new tRNA sequence, the amino acid, the previous amino acid, is going to be linked to the chain. Then we move on down the line, and in order, and it's really important to know this, that this all was specific from the original DNA molecule. So the order of the amino acids is specific for the original gene sequence. Down the road, we can talk about the specific job or the destination of where these actual proteins, the finished proteins, would go. And of course, it has a lot to do with the signaling that goes on in the cell. But these finished proteins are going to have a lot of different functions, therefore they're going to go to a lot of different places, whether they are a hormone, an enzyme, perhaps a pigment, perhaps a structural protein. There's a number of different functions that these proteins can have. Just remember that they are going to determine phenotypes of the cell and thus the phenotypes of the organisms. There's a few labels here on this diagram that are beyond the scope of us, some things that shouldn't look familiar to you necessarily, but the question remains the same. Can you tell the story? Simply put, could you, in a written form, describe the central dogma of life? How we go from a gene in the DNA molecule to a finished product of a built polypeptide and finally a protein by ordering amino acids in a specific sequence. A quick note here is that we need to know is that the building of a protein is a little different in prokaryotes. You know, remember, there's no nucleus in a protein, or excuse me, in a prokaryotic cell. And straight from the AP Bio curriculum, we need to know that in prokaryotic organisms, transcription is coupled with the translation. So that means that they happen together. Translation involves many steps, including initiation, elongation, and termination, but it's going to occur a little bit differently in prokaryotes than eukaryotes. So you want to pause here, and these are all good points to identify, differentiating between the building of proteins in prokaryotes versus eukaryotes. So with no nucleus, or obviously not going to be a nuclear membrane to separate where the DNA is from the RNA. So DNA is going to be found in the cytoplasm in prokaryotes. We don't need to edit the mRNA because of the fact that we don't have coding regions of RNA and non-coding regions of RNA like we do with eukaryotes. And therefore, the ribosomes are going to actually read the mRNA right as it's being transcribed. So transcription and translation are going to happen together. So take a minute and you can pause and identify the differences between prokaryotic and eukaryotic transcription and translation. So there you go. Here we are with the same diagram that we just showed you moments ago without any of the labels. Can you tell the story of going from specific segments of DNA and how we can transcribe that into the RNA message that can then wind up at the ribosome, and the correct transfer RNA is going to bring the specific amino acid at the right time so that we can build a specific polypeptide chain that is going to eventually code for our phenotype. In the words of the master, I hope that helps.